Thanks everybody for the opportunity to present the AdAlta story to you and the investment opportunity. I'll be making an introduction to the company and a bit of an overview and a summary of our strategy, and then handing over to, as David said, our new Managing Director, Dr. Tim Oldham, who will take you through the detail, and then we'll jointly answer questions at the conclusion should you have any. So some of you in the room I know are familiar with the AdAlta story. AdAlta is a listed Australian drug discovery company commercialising a new class of protein therapeutics. These drugs that we're developing are neither a traditional chemical drug nor a traditional antibody drug. They fall somewhere between the two and have a very interesting set of therapeutic opportunities as a result of that are quite differentiated from both of those other drug classes in terms of being able to hit drug targets and diseases that have traditionally been quite inaccessible. Where's my... This changes every year. Green one? Oh, sorry. So the investment summary from a high level is we have a drug platform in terms of these antibodies that is able to generate multiple products against multiple disease states and indeed multiple diseases within our existing molecules target disease state as well in terms of different fibrotic diseases and different organs. Our lead internal program, AD214, many of you will be familiar with. It's been in preclinical development for some time now. We're now currently completing the last of those animal studies that will then enable us to move into the clinic and for first time in man studies next year, which is obviously a major inflection point and value driver for the company. We've received a US orphan drug designation for our initial molecule and disease, which is idiopathic pulmonary fibrosis. And we have a number of collaborations both in the past and at foot at the moment that provide some level of commercial and external validation from multinational companies in terms of the attractiveness and the commerciality of this drug class and this approach. The company raised money through the course of calendar 2019 and is financed to meet the milestones that we have been outlining to the market recently and in the more distant past. We have a group of shareholders that is a very healthy mix of institutional investors, which have been supporters of the company for a long time. We'd also have one major institutional investor that came on board additionally to those at our last fundraising. So very strong institutional support, but also very strong retail support as well. The value drivers for the company and the strategy for the company we've outlined before. Tim will go into this in more detail and we'll be outlining and emphasising this to the market more strongly in the future. We have an in-house drug pipeline development program that's focused on our lead candidate 214. 214 is a version of a molecule that we've been working on for some time that's been formulated specifically for this disease state. So we have data around 214 and its predecessor 114 in a number of disease states hitting the same target. As I said, our lead program is the idiopathic pulmonary fibrosis program, but we also have other preclinical data in organs such as the kidney, so fibrotic diseases of the kidney, and we also have some preclinical data in the eye, in ophthalmic indications. The company also has a pipeline of other molecules targeting other difficult to drug targets. These have been outlined in broad terms in terms of the molecular target we've been looking to access, and we'll be expanding and explaining that further going forward as to which disease states are going to be most attractive for those targets, and hence we can start to outline commercial opportunities for those other molecules and newer molecules that we'll develop. And the third strand of our strategy is to look for partners and commercial validation for our class of chemicals. So we have had a number of academic collaborations in the past. We have had a successful relationship with Roche in the past over one of our earlier molecules, and we recently announced a collaboration with GE Healthcare, one of the largest diagnostics companies in the world, to co-develop diagnostic agents against novel cancer markers for them. And with that, great pleasure to hand over to Dr. Tim Oldham.
Thanks, Paul, and it's a pleasure to be here in my, first public outing as the new CEO at AdAlta. Yes, I am one of the Faulding alumni that David referred to a little bit earlier. I joined Mayne Pharmaceuticals, as it then was, about a month after it acquired Faulding. Worked alongside Mike and many of the other folks you've heard from in this sector along the way. But I've always been working in some of the high growth and more complicated parts of that business and business's sense. Starting with biological or biosimilars as they then were when they were just a twinkle in the eye of the European Medicines Agency, working through development of the first biosimilar products, then finishing more recently in the cell and gene therapy and the biospecific antibody arena before joining AdAlta. So AdAlta is a great little company that, for me, gives me the opportunity to deploy a whole range of skills in complex product development, alliance management, business building, strategy, and corporate leadership that I've developed over a number of years. But more importantly, it's a very, very cool little company with some very exciting technology. This is not just another drug discovery platform of a Me Too variety. The underlying technology has a genuine potential to develop drugs against some really difficult to target areas that have been intractable to traditional drug development techniques. We also have, as this slide illustrates, multiple ways of generating revenue and value. This is not a one-product company. This is the potential to build a sustained company that not just creates sustained value for our shareholders, but hopefully also generates significant employment here in Australia. And finally, it's approaching a value inflection point. We'll have our first product in the clinic early next year, which is obviously a huge milestone for any early-stage biotech company. So I'll start talking about our platform in a little bit more detail. What is it that we do? We call our platform antibodies. Essentially, these are a protein drug, but essentially they have unique characteristics in that the binding domain, let me cycle back a little bit. An antibody binds very specifically to a thing we call a drug target. A target is a biological structure that regulates something that happens in your body, be it cell signaling or a metabolic process or something similar. Antibodies bind very specifically to those, as opposed to small molecules, which tend to be more general simply because they are very small. So with small molecules, you have the risk of off-target effects. They can attack multiple different kinds of receptors, which is why antibodies were so successful, because suddenly we could get specific. The problem with traditional antibodies is we can't always get them where we want them to get to. And our antibodies are unique in that they were developed from an observation in sharks of all species, that they actually have a much longer binding and flexible binding domain on the end of their antibodies. And we've taken that, humanised it, and that means we can now, from our library of 20 billion different variants of this structure, find targets to bind to receptors that are difficult to target with traditional antibody mechanisms. So essentially we're trying to drug very difficult to drug targets, and the reason these have been hard to drug is why the diseases that we get to as a result have high unmet needs, because we simply haven't been able to develop drugs against them. So our antibodies are a new approach to the treatment of a wide range of human diseases that haven't been able to be drugged previously. And importantly, we can use them just as the antibody, or as in the case of AD214, we can actually couple them to other structures in order to create a more pharmacologically suitable drug. So this is our pipeline, and really this highlights the diversity of applications and revenue models that we can generate. Our lead product is AD214. As Paul mentioned, we are in the final stages of the IND enabling studies and manufacturing to enable us to take that into the clinic in a disease called idiopathic pulmonary fibrosis. I'll talk about what that means in a little bit in a moment. But it'll also go after a whole range of other fibrotic indications, and I'll show you a little bit of data about that. So one product, multiple indications because of the receptor that it targets. But you can also see in the target column that we've got two other products where we're starting to develop or discover antibodies against new targets. And don't worry about all the acronyms. Biologists seem to love these, and they're totally confusing even to those of us who have been around for a while. The point being that they're different sets of acronyms is a different target. And finally, the bottom two lines illustrate our ability to partner to increase the leverage of our platform. The most recent one, which again I'll talk about later, with GE, announced just a couple of weeks ago, is to find antibodies against targets that GE would like to use as diagnostics. Now, we couldn't and wouldn't pursue this application ourselves, but by doing this we can generate revenue in the form of milestones, potential royalties down the track, and additional development activity that enables us to further validate the platform. So this is a really important way of starting to generate momentum behind our technology. So let me talk about AD214 for a moment and fibrosis in general. So all of us would have had an injury at some point. And the first thing that happens when you cut yourself or graze yourself or any other injury of that nature is it will inflame as the immune system swamps in to try and clean the wound up, and then it will scar. And the more the inflammation, the more the scarring. And the scar is rigid, stiff, and not very flexible. So we're all familiar with what happens when you scar on your skin, but imagine what that happens when the scarring, the inflammation, and the resultant scarring happens in an internal organ, like the lungs, which flex enormously about every second or two. If you suddenly start having that scarring, which is called by the medics fibrosis, then your lungs stiffen up and you can't breathe. Similar thing can happen in your kidneys. Similar thing can happen in your eye. And a disease called macular degeneration. A similar thing can happen in the liver. All of these are examples of what we call fibrosis. And all of these, if they occur, can be debilitating. And I'll talk about the lung example in some more detail. So AdAlta's 214 is designed to treat and reduce and manage fibrosis. And we think it can potentially have application across all of these different indications, but our initial focus is on the lung. So this gives you an example of the potential impact. These are in animal models of our lead antibody. So the antibody is the binder on the top of AD214. We used to call it AD114. We then coupled it to another structure to produce 214 to improve its pharmacological properties. But on the left-hand side, you can see a normal lung tissue of a mouse. On the centre image, we've treated the mouse to create fibrosis. And the blue staining goes to collagen, which is one of the major structural features of fibrosis. And so you can see that all those pores that could hold air have closed up, and the oxygen and the air transfer capacity of the lung has shrunk enormously. On the right-hand side, we've then treated that mouse with our AD214 or AD114 antibody for 21 days, and you can see that the level of collagen is reduced significantly and the structure and the architecture of the tissue has improved enormously. So it's this kind of data that gives us the encouragement to progress this drug into patients. This is an example in kidney. Again, we've created a kidney example. So on the left-hand column, you can see healthy kidney tissue, again in a mouse. We use folic acid to initiate fibrosis in the mouse kidney. And in this example, the top row, we've used our technology in a preventative or prophylactic mode. In the bottom row, we've used it in treatment. So in the first row, the AD114 was administered at the same time as the folic acid. So the damage occurred at the same time the drug was present. In the bottom one, we allowed the fibrosis to establish and then treated with AD114. The middle column is the negative control, so an antibody that we know doesn't work, and we can see that it didn't make a lot of difference. The second from the right column shows the impact of AD114, and you can see in both preventative and therapeutic modes that the level of blue staining has gone down significantly. And the right-hand column, we simply compared that with one of the other antifibrotic drugs in development. And again, we believe that there is signs here that we can be better than some of the other drugs coming through the pipeline. So why is this opportunity so interesting? There are 300,000 people around the world today living with idiopathic pulmonary fibrosis. Idiopathic simply means we don't know exactly what caused it. 40,000 people die. The average life expectancy from diagnosis is under 4 years. There are only 2 drugs on the market today for this. They were first approved in 2014, and prior to that there were no treatments. In 2017, they generated around about 1.5 billion in sales, and that's forecast to increase to 3.5 billion by 2025. And these are drugs that work in 30% of patients. They don't reverse the disease, they merely slow it down. So there's still enormous potential for improved therapies in this space. So the benefit of those 2 first approvals is that people suddenly got confidence that this was a disease that could be treated. Prior to that, it was in the category of too hard. Well, now we know it's not too hard. De-risk number 1 for AdAlta. The second one is there are a number of products in development. So we're not making up the clinical pathways. It means that we're likely to have less backwards and forwards while we do a range of different studies, because the studies have already been worked out with the regulators. And finally, we're targeting a novel target. We call it CXCR4. I won't go into the details, but essentially it's different to the approaches that the other drugs in development are taking. And most of the reports I've been able to read so far suggest that there is going to be a place for, given the complexity of a disease like fibrosis, a place for multiple mode of action drugs to be used in combination. So where are we up to? We're completing final toxicology studies at the moment. That data is actually due in our hands later this month. The result of that study will enable us to finalise the clinical trial, the design of our first clinical trial. We are also in the middle of manufacturing the clinical material for our first clinical trial, which we expect to start early next year. You'll also see on the bottom of this slide the times at which 2 major recent deals were done in this space between biotech companies and larger pharmaceutical companies for IPF drugs. And the point being that they were made really, really early, and I'll show you what they were worth later on. So the second revenue stream that we have is licensing our platform to other companies to enable them to do drug and diagnostic discovery. The GE Healthcare licensing deal that we executed a couple of weeks ago is an example. And in this case, we will screen our iBody library for targets that GE are interested in, the first of which has been announced. And they pay us a series of milestones and the cost of doing the research to pan that library. If the development is successful and ultimately leads to a product, then there are further milestones and royalties that come along. The important thing for us is not just the revenue, but the potential for us to actually validate further that our platform produces drugs in a whole range of different settings. So this is an example of the value of different aspects of AdAlta's technology. The first row shows some of the recent deals in fibrosis. The Adheron deal on the left and the BridgeBio deal on the right are the 2 that were on the previous slide. And you can see that for very early stage assets, only in phase 1, we're talking double to triple $1 million digits, milestones, and $1 billion potential milestones through further development milestone successes. Now, I'm not suggesting that we can deliver one of those deals during our phase 1 trial. It'd be nice if we did. The point of this is really to emphasise the level of interest from Big Pharma in these kinds of products. We're operating in a field which could be broadly termed microantibodies. There are a variety of different ways of trying to produce an iBody-like structure. And the second row shows the value of some of those deals, deals that have done off platforms like that. Again, similar structures of deals at similar stages of development. And companies like Sanofi, and in the case of Ablinks, actually acquiring the entire platform because of the value it brought to their entire drug delivery effort. And the final row is labelled deals in G-protein coupled receptors, GPCRs. This is one of our first targets, including the fibrosis drug targets at GPCR. And again, this is a hot topic for big pharma companies, largely because these receptors are a key pathway for multiple different cell signalling mechanisms and therefore implicated in many, many diseases. So these are the types of transactions that we're aspiring to, and we'll be working very, very hard to build our company to earn these types of relationships with big pharma companies. So in 2019, it's been a big year. We successfully completed the cell line and the manufacturing development for AD214 and are currently manufacturing the clinical drug substance. We've commenced the phase 1 enabling non-human primate studies, and as I mentioned, the final report is due imminently. We've entered the partnership with GE that validates our ability to partner the platform and develop that second revenue stream. We've continued to develop and broaden our pipeline through a research agreement with Accelerate and further publications. The board's skills have been expanded as we move into the clinic with the appointment of Dr. Ros Wilson, brings some clinical expertise to the board, and my appointment last week, which is probably well, I'd like to say it's significant. You'll tell me later on whether it's been significant or not. So what's coming? This half we committed to announcing a partnership agreement, Tech, GE done. We committed to publishing some key iBody data. That has been done, and we've got more in the pipeline. We have still coming the toxicology study, and in the first half of next year, obviously the big event will be the phase 1 human clinical studies commencing. But we'll also undertake to provide an update on strategy, the pipeline development and the broader corporate strategy, which I would imagine you would all expect as a result of me joining. The management team is deeply experienced in protein discovery and protein manufacturing through Mick Foley and Dallas Hartman. My own experience I've outlined already. And our board and scientific advisors are really hands-on. So I've highlighted on this slide some of the companies they've all worked for or worked with in the past, because it's their expertise that in a small virtual company is actually mission critical to ensuring we get access to the right experts, either directly from them or through their contacts, and they've been invaluable to the progress of the company to date. So in summary, we're a platform technology that can develop multiple pipeline products and partnerships. We have a lead asset about to enter the clinic for the first time. We have additional expansion opportunities through partnering. We have cash to get us into those phase 1 clinical studies for AD214, and we have an experienced leadership team ready to deliver. Thank you very much.
Thanks, Tim and Paul. Can we have perhaps a few questions? We've got a question over here.
Thanks for that. That was a great presentation. I just wanted to ask about first of all, I want to say, Tim, happy birthday. This is a very special birthday day, I just found out.
Thank you, Angie.
With the phase 1 trial, like how many in that trial and where you're doing that?
There's, in terms of total patients, there's 14 patients. They're healthy volunteers, and that study's most likely going to be done at CMAX.
Great, thanks.
Can I just follow up from that question?
No problem.
Hang on, there's a mic here.
How long will those the phase 1 study go for, and when do you expect
I'll let you take that one.
results out of that?
I'll let Tim take that one.
So the study itself, we can't nail down the exact design until we get the final results of the toxicology study, because that will set dose. It'll set the number of cohorts we actually need to do, and it will also determine exactly what additional studies beyond the pharmacokinetics we do. But we're currently expecting that study to take around about 3 months, with another 2 to 3 months for the report write-up. So we expect the results of that sometime around the middle of the year. Yeah.
Some more questions? Got one here.
Hi, Tim. My name's Tim Elliott. I'm a gastroenterologist. And I see patients with Crohn's disease quite frequently who suffer from an inflammatory condition of their bowel. And as a consequence, they develop a lot of fibrosis with chronic inflammation. And that causes lots of problems, need for surgery and that sort of thing. I just wondered whether you considered this drug in the context of Crohn's disease.
I'll take that. So that, for reasons of animal model complexity, quite frankly, more than anything else, isn't one of the disease states that we've assessed with our molecules to date. I was about to say my gut feel. That would have been a very bad pun. most likely it would be effective in that disease state, but we haven't got any data to confirm that. But we've assessed it in a number of different fibrotic models. More than has been presented here, and it's been effective in all those fibrotic models. Crohn's disease is just a really tough animal model.
And I guess adding to that, there are drugs in this class, or people trying to develop these kind of drugs, that are starting to think about Crohn's disease as well. And it does fit into that category where the existing therapies have some pretty unpleasant side effects as well, obviously, which discourages continuance of therapy, which creates the ongoing unmet need.
I've got a question, Tim and Paul. Can you just tell us a bit more about Mobazil and what is known from that's effect on the particular receptor you're targeting, and whether there's any insight from Mobazil as a target, even though it's a stem cell, also a stem cell
Sure.
mobilizing drug?
So this is a really good example of why our approach is important to developing drugs against these kinds of receptors. The drug David's talking about is another drug that targets the same CXCR4 receptor, and it has the effect of mobilising your blood stem cells. Essentially mobilises white blood cells to be produced and migrate from the bone marrow into the blood in response to infections and various other things. The drugs that cause, and Mosabel is a drug that has been designed specifically to encourage the mobilisation of white blood cells. It doesn't actually have any effect on fibrosis. It goes after the same receptor. So it's a good example of how one receptor, depending exactly how you engage it, can have different effects. For us, it was obviously one of the first things we wanted to know was, well, which bits of the receptor are we activating? Are we actually causing mobilisation of white blood cells, which wouldn't necessarily be a good outcome in the course of treating fibrosis? And so there was extensive preclinical work done to demonstrate that no, we don't actually mobilise white blood cells, and that's an endpoint that we expect to see coming out of the tox studies as well.
So you're expecting a better safety profile than, say, Mobazil?
Different.
Yeah, it'll be different, right? So you essentially you've got one. Think of it as the receptor as an intersection, and a drug coming down to the intersection can cause the cell or the body to do two different things. Well, Mobazil takes it that way, and we're going that way, so it will be different, not necessarily better. Well, it will be better than Mosabel at treating fibrosis, definitely.
Yes, good.